Yes. In this meeting, I'm presenting updates on our big single-cell sequencing study of tumor and immune cells in patients with MGUS, smoldering multiple myeloma, as well as overt multiple myeloma. We have performed single-cell sequencing in 879 samples from 517 specimens, both bone marrow and peripheral blood, and 362 individuals, spanning all stages of progression from MGUS to myeloma and including healthy donors. For a large group of individuals, we actually sequenced both their tumor cells and their immune cells to enable integrative analysis of tumor and immune biology. And on the tumor fraction, we performed single-cell B-cell receptor sequencing, approximately 370 libraries, and on the immune fraction, 350 libraries of T-cell receptor sequencing as well. So a total of 1,600 libraries. It's truly a massive effort and the largest in the field right now. We have approximately 5 million cells after filtering, probably double that before that. And so using this massive data set, we have been able to gain tremendous insight into what we call the myeloma immunity cycle. So we see with progression increases in cytotoxic T cells, particularly terminally differentiated ones that express granzyme B. And so one of the questions that we have been dealing with, and this is sort of part of the update that I'm presenting in this meeting, is how can this be? Terminally differentiated cytotoxic T cells are actually known to contribute to anti-tumor immune responses. So how do we see them increased in patients who actually progress? So what we see here is that cytotoxic T cells clonally expand and terminally differentiate, and that explains the increased numbers we see in patients. And they also proliferate more, and they also express higher levels of interferon gamma, all paradoxical given the association of these populations with the risk of progression. So our hypothesis is that there may be a confounder explaining this phenomenon or something else may be going on. We show again and again, though, that it's actually not confounded, for example, by chronological age. As individuals age, they do show more clonal expansion in cytotoxic T cells. We do not see that be the case here. While, obviously aging does play a role, the progression of disease has its own distinct effect on these populations. So our hypothesis is that despite these seemingly advantageous changes that we see with progression, the T cells are actually dysfunctional, and a lot of the data I'm presenting today actually speak to that. We show that there is a decrease in the expression of a T cell functionality program that actually has protein level correlates. We see that those T cells, while they appear to be functional, they are actually enriched for terminally exhausted T cells. So there's certainly T cell dysfunction going on with disease progression that likely contributes to the loss of immune control of tumor cell growth, and therefore these patients are more likely to progress.